everyone. Thank you so much for coming. We're so happy to have you here. The Hellenic Innovation Network, with the support of the Greek Consulate in Boston and MIT Enterprise Forum Greece, is driven by a group of people located in Boston and Athens who meet monthly to organize events that are held in Boston to educate those in the United States about what's happening in technology and innovation in Greece, and in order to provide the opportunity for Greek entrepreneurs to come over to Boston and pitch and network within the very rich Boston tech ecosystem. Under the leadership of George Demosthenes, we also have a CEO group which meets every month. And while our events are normally held in Boston, during this period of time, during the COVID situation, we are organizing these fireside chats in order to keep our community connected until our next in-person event, which is scheduled for November 12th at the MIT Media Lab, and I hope you will join us then. In the interim, stay tuned with news and events. You can check us out at hellenic.org, where you can also register. And if you have any questions during the event today, please feel free to participate and share your name, and they will be addressed at the end. So without further ado, I'd like to introduce you to the Consul General of Greece in Boston, Stratos Efthymiou, who has provided incredible support to us during this period. Thank you so much.
Thank you, Marina. Hi, everybody from the Consulate General of Greece in Boston. We are very happy to support and be actually an integral part of the Hellenic Innovation Network, a great effort which is very complementary to our own vision for being a science diplomacy hub, facilitating scientists, innovators, connecting together the innovation ecosystems of Boston and Athens. The Boston ecosystem has obviously been playing a very central role globally in research and innovation, especially right now during the biotech battle against COVID-19, a crisis which Greece has tackled so successfully so far, letting science shape public policy. Of course, the issue of antibody detection is of critical importance for the restarting of our economy, education, tourism, of our life in general. And I'm looking forward to the conversation between two exceptional Greeks in biotech, Professor Alexopoulos and George Petrocheilos. With this, I pass it on to Vasilis Papakonstantinou from MIT Enterprise Forum, Greece.
Thank you, Strato. Hello, everybody. Thank you for joining this webcast today. MIT Enterprise Forum Greece is a non-profit organization with a mission to support technology entrepreneurs turn their startups into global companies. Our flagship program is the Startup Competition. We are currently in the final stage of the competition's 6th edition, with great teams, some of which are coming straight out of the Greek universities. You can follow us on social media and keep an eye about the finals that will take place in early July. We are particularly interested in ventures coming out of the Greek universities, so it's a great pleasure to welcome guest speakers like Leonidas Alexopoulos. Before handing it over to have this discussion, I would like to welcome also our moderator, George Petrocheilos, who is a managing partner at Nexus Management Capital, which he co-founded back in 2016. He makes investments in early-stage biomedical technology companies founded by world-renowned scientists and entrepreneurs. George sits on the board of Hellenic Innovation Network and is also an advisor to MIT Enterprise Forum Greece. So without further ado, George, the floor is yours.
Hi, guys. Let me make sure I figure this out. Hello, everybody. Thank you for joining us. And thank you for the introduction, Vasily and Marina and Strato. I'm very excited today to have the pleasure to talk to Leonidas Alexopoulos, who I got to meet recently, but I'm extremely impressed with his background and his career. Leonidas is an associate professor of mechanical engineering at the National Technical University of Athens, where he's the director of the Systems Bioengineering Group, focusing on systems biology and medical devices. He also is an entrepreneurial professor, having co-founded Protavio in 2012, a biomarker discovery services company that we will talk about today. So Leonidas studied mechanical engineering in the Aristotle University of Thessaloniki, and then came to the US to get his PhD in biomedical engineering at Duke University, one of the best programs in the country for BME, and continued with postdoc degrees at Harvard Medical School and MIT, or actually vice versa. MIT first and then Harvard Medical School. Leonidas's research combines mathematical modeling of biological systems with novel experimental technologies for early drug and biomarker discovery. So with that, Leonidas, thank you, and welcome to the Hellenic Forum. So thank you for being with us. Hi from
George.
New York. I know you're in Athens. So
yes,
great.
it's a great pleasure to be here, and I look forward for this exciting discussion.
Absolutely, thank you. So let's start. So today, the way that I'm thinking about our conversation, after this introduction, I want to talk for a little bit about Protavio to put everything into context based on your research and the commercialization of it into Protavio. Get to the topic of the day, COVID-19 antibodies and detection, and then get back to Protavio to talk a bit more about the business development there and the general climate in Greece in starting companies and specifically spinning new codes out of labs in academia into successful companies like Protavio has been. So with that, as I said in my intro, Protavio is a biomarker discovery services company. Tell us more.
So, as you said, I'm a professor at the National Technical University of Athens, here in Athens, and I came back to Greece when the crisis started. So I came in 2008 to Greece in 2009.
Timing.
Yeah, perfect timing. So I was for 10 years in the US. I loved the US, and it was a great place, great people, great network, and I saw how you can combine good research with entrepreneurial like spirit. But I thought my time came to come back to Greece, and it was a tough decision. I can say that after 10 years in Greece, I didn't regret it. I really liked that I came back to Greece. I had the good opportunities to follow academic careers in top universities like Imperial College, but I decided to go back to Greece. For someone else would have thought that it's better to stay like in a top tier university, like world-known university. But I really feel that I did something very good that I came back. So a few years later, I started the lab in the university, and a few years later, in 2012, as you said, we started Prota@Once. Prota@Once started by myself and Sofia Stamatatou, who's the co-founder of Prota@Once and Protavio. It was a very hard time because if you remember, like back in 2012, Greece was full of troubles. You couldn't find venture capital, you couldn't raise money. We started just with 5,000 euros by borrowing from what we call FFF, Friends, Family, and Food.
This is great, and I think this is a very impressive story. I don't want to get into that now. I think I have many more questions on that on the third part because I find this, like I said, extremely impressive. Why don't you tell a little bit about what Protavio does so that our audience understands specifically what the company is offering today?
So we're a biomarker. Protavio is a biomarker discovery company. So we find
What does that mean?
We find good proteins that can be like a diagnostic tool for COVID-19, for a cardiovascular disease. They can separate patients to who's going to respond or who's not going to respond to a drug. So we have this multiplex technology to be able to identify a good biomarker. And if there is a good biomarker, we have all this pipeline, the GMP, good manufacturing practices, or the ISO 13485, this legal framework that is required to make this biomarker an assay, a kit that can be used from a diagnostic lab.
And can you talk a little bit about the customers that a company like yours has? today?
So when we started our first customer. It's something that is public, so we can share this thing.
Sure.
It was with Philip Morris. So Philip Morris was our first customer. It was a tobacco company, and after that, a lot of our customers we have all the tobacco company, all the tobacco
Why
and
so?
cigarettes.
Why
Sorry?
so? Why?
They are interested in assessment. So it's not a drug assessment, it's a cigarette assessment, but it's exactly the same kind of assessment. You develop biomarkers, you do multi-omics data and data analytics, and you were able to identify if something is toxic or non-toxic. So you can compare regular cigarettes with electronic ones, or you can use it to identify among all different kinds of electronic cigarettes which one is less toxic.
Very
So
nice.
that's how we started, but later on we expanded in major pharma companies. We have major collaboration with big pharma companies, with academic labs, and in the last 4 years, because of this, I mean, we were researchers back then, and I was pure researcher. Sophia has an MBA, and she's a chemical engineer. So. But we hadn't been exposed to what is the regulatory status. But in all those last 5 years, we were able to understand what the market needs, and we established this pipeline, the regulatory pipeline, to develop IVD quality assays.
And just so everybody understands, I know we talked about it. These are clients all over the world. These are not clients just in Greece.
We are a 100% export company. In all these 9 years, our turnover from Greece is 5,000 euros. It's a 100% export company.
Very nice. So let's talk a little bit about your testing. It's protein-based, right?
Yep.
So I have a question. When we introduced each other last week, we spoke a little bit about one of my portfolio companies, Thrive, earlier detection, which is basically a screening test for early pre-metastatic cancers. And Thrive uses a protein-based testing combined with nucleic acid sequencing in a biased way. Okay. My question to you is, how do you integrate protein-based techniques such as next-generation sequencing? Protein-based techniques with unbiased genomic testing, such as NGS, next-generation sequencing, for the purposes of this type of treatment.
Yeah. So at the discovery phase, you can do all these kind of things. We have the computational tools, the in silico analysis, that allows you to combine multi-omics data. This is a combination of multi-omics data using machine learning, artificial intelligence, this kind of analysis. And we can combine multi-omics data. But when you offer a kit at the end of the day, when you offer a product, a diagnostic kit, usually you don't combine those two. So you combine it as a service. So when you would like to assess tobacco, or you would like to assess a drug, if it's a toxic or if it has efficacy on the clinical trials, and you need both worlds. But at the end of the day, when you would like to put a product on the market, usually the product is going to be either a protein or it's going to be a DNA.
Thank you. And as switching to our main topic, COVID-19, as you know, all of us in life sciences got really busy at the beginning of March and had to reassess a lot of things, all the way from our technology, our offerings, to balancing budgets and creating contingency plans. Although most of my investments are in the US, I'm sure this applies to Greece and to everywhere in the world. Every one of those life sciences companies, if they could have some applications to COVID-19, they try to add that to their offerings. I have companies that focus on detecting sepsis, for example. They added on their platform ways to detect respiratory failure, which is more relevant than ever now with COVID. And a lot of companies on the medical device side in the US, or the blood shortage space that I have, are focusing on applying to emergency usage for the FDA. Blood shortage, big issue right now with COVID, including America. Talk a little bit about what you and Sophia thought about when this started in Greece. how did Protavio act? Because you have a unique positioning to focus on this detection side.
So for us, it was not something new. So for us, we had this GMP pipeline for all these kind of assays are called serology assays. So what you would like to do is you would like to measure antibodies. Yeah. So for these antibodies, you put like an antigen on a surface that can be like, in our case, it's a bead, but it can be like a 96-well plate. And then you mix it with the blood, the serum, and you would like to see if the antibody goes and binds to that piece of COVID or to that piece of virus that you have on the surface. So we had this project because we had started this pipeline, and we had collaborations with public agencies abroad and with big pharmas. So we had this pipeline. For us, it was a pipeline that was ongoing. We have been audited and was working well, and it was GMP and ISO 13485 compliant. So it was not a brainer to reposition this pipeline and says that instead of having the vaccine thing, because we were doing it for vaccines, instead of having that particular vaccine on the surface, I'm going to put a piece of COVID and I'm going to develop it. And that's what we did, but we did it on a multiplex format.
I see. So there's a couple of methods of doing this. Which one is your method, and why, which you kind of touched upon, why is it different? And I'm sure you would say it's better from other methods.
Okay, I'm not going to say it's better then. So there are many different kinds of ways to be able to measure the antibodies. And it depends on the user. It's different if you are a screening center. It's different if you are a person in somewhere far away that wants to get an answer right away. So there is this lateral flow devices that, like the pregnancy strips, that they offer this kind of yes/no answer. And there is the other ones that is for much higher throughput, that is based on, it belongs to a lab. And our method is the second one. So it's a high throughput. But the main difference compared to the other methods is that it's multiplex. So we can measure a lot of things simultaneously in parallel. So instead of measuring only one part of the COVID, of the virus, we measure 3, 4, 5 different parts of the virus, plus old viruses, the common cold. So we can have a panel of 10 different things, and we can have much more certainty that the assay works well, because you're not based on one measurement, you're based on 10 different measurements.
That's great. Talk a little bit about the challenges of creating such a diagnostic antibody test for COVID-19. We've all read and we all have seen there is a bit of controversy surrounding the accuracy of these tests. What do you think? Let's talk about accuracy.
Yeah, so all those methods have sensitivity, specificity, and a combination of those two that is called accuracy. So it depends who's the end user. So let's say a pregnancy test, right? I am developing a pregnancy test, and I'm testing this pregnancy test. It's a fake test. It always says, no, I just put water in it, and it's a fake test. So I put I make this test, and I test this test in 1,000 women. And I go out on the public, and I get 1,000 women, I take this test. 10 of them, they're gonna be pregnant at this time, and the test is gonna say no. So this test has 99% accuracy. It's a perfect test. It's a fake test, but it's a perfect test. So you can play a lot with the numbers, and you can say accuracy 99%, but 99% doesn't mean anything if you don't understand what exactly accuracy is. So our test, so that's why there is the specificity, sensitivity. One refers mostly the sensitivity, how well can detect someone that has COVID. So if I'm a person, an end user, and I see the test and I see that the result is positive, I really want to know that when I see the positive result, this is true, and this is sensitivity. Or if I'm a screening center, maybe I don't care about that much, so much of that, but I care that I'm going to screen 1 million people, and out of the 1 million people, the prevalence, like for example in Greece, the prevalence is like 0.1%, 1%, something like that. If my test has 5% error, which is not bad for a test, I'm going to have out of the 100 people, 5 people that's going to be positive. But out of these 5 people, the true positive is going to be only 1 because the prevalence is 1. So that's why there is all this kind of debate if the test is good or not. All tests are good, but you need to know, you need to read the specifications, you need to understand the statistics, and you need to apply each test for the right case.
Thank you. It sounds like you're extra busy these days with this. So let's go a little bit back to Protavio. We have 5 minutes to talk a little bit about this and the general landscape in Greece. Tell us a little bit about, we touched upon it very briefly at the beginning. You started in 2012. How did you meet with Sophia, your co-founder?
Okay, that's a different story. Okay.
I know the answer, but I just think the audience would like it too.
I know Sophia when we were, as kids, we were playing together from Ioannina. We're both from Ioannina. We're from the same village in the middle of nowhere. The village is called Paliopyrgos. So we were in Paliopyrgos and we were hanging out together in Paliopyrgos. All these years we knew each other, we were keeping in touch, we were very, very close friends, we were appreciating each other a lot. When I moved back from the US, we were just hanging out a lot. She's a chemical engineer and she has a master in automation and another master in business administration. I was more on the technical side. She was working for a big dairy company. And talking to each other, we decided to start this company. Later on, we got married, and now we have a 3-year-old kid.
Very nice. Congratulations. So you started in 2012. It's 2020 now, 8 years. Are you guys still together with Sophia?
Yeah, of course. She's right next to me now. She's
she likes the question. Good. So in 2012, when you guys started, did you talk to any people like me? Did you call up any VCs? And I know the answer is no, but is it no because you didn't feel like dealing with it? There were too many options and you didn't know which one to choose, or simply it was a really bad time?
It was a very bad time. There was no options available. You couldn't borrow money from the banks. You couldn't get a VC. We wanted to have a VC actually because we were broken. We didn't have any money. Yes, at that point, if you remember in end of 2011, beginning of 2012, in Greece it was horrible. The center of Athens, Syntagma, was burning down every other day. We talked to some external VCs, but no one wanted to invest in Greece at that point.
Out from abroad?
Outside from Greece.
Yeah.
Yeah. And
so how do you find the first 5,000? Your own money?
From my grandma, my father, my aunt, a little bit of my money, and a little bit of.
And today, so people have into perspective, the company has grown. I'm not sure if you want to disclose how big the company is, but it has done very well. Leonidas and I spoke about this the other day. So congratulations. This is very impressive. How much of the company do you guys own?
100%.
I just wanted you to say that. That's very good. You know, sometimes persistence gets rewarded with that.
Don't get me wrong. We didn't, it's not, I mean, at the end of the day, of course, it pays out and it's good right now, but it was very hard. If I had to do that from the very beginning, I wouldn't have gone this way. And the chances of failure were high. So
for sure. And that gets to my next question. VC in Greece today. One quick comment on the general landscape. Don't just focus on biotech. I know there's been a bunch of groups the past couple of years that have raised significant funds with the help of the European Stability Fund and all the other networks available. Do you have any interest in working with any of those people? And to get it more specific back to life sciences, do you think there are groups right now in Greece that have the expertise to help scale a company like Protavio?
Okay, very, very good points. So either you are Protavio or you're another startupper in Greece, it's a great time to start a company. It's really, the environment is very good. the company needs to be extroverted, needs to have like a footprint somewhere outside.
Correct.
And of course, a VC funding or somewhere like a network that brings the inside of Greece, brings it outside, like MIT Enterprise Forum, like the Hellenic Network, like many others, Hellenic Biocluster. You can link to all these networks and you can have a footprint outside, including VC outside. So in Greece now, it's a great time to start a company. There is a lot of VCs available. And it's not like how it was like 5 or even 8 years ago.
Yeah. How about life sciences? How about companies in our drugs, devices, diagnostics market?
Usually these companies require significant amount of, and it's not that mature in Greece. Greece is a small, it's a small country, and you should not be based on the Greek economy. So maybe some like
I agree 100% with that.
specialized funding, like your fund or like some other funds that they are available, gives you this network that you require to start a company and get like a footprint outside of Greece.
For sure. One more question in relation to that, and then we will open it up to every one of you listening, so that you can go on the YouTube channel and start posting questions. We're a bit off time, but I want to ask this. As you know, my sole focus, Leonida, is to find and work with people like you around the world that are professors, have a primary academic appointment in the world's leading academic institutions, and who also think entrepreneurially, who can also historically have translated their basic research into commercial applications. Like you are doing with Protavio, with your primary academic appointment at the National Technical University of Athens. In the US, we have seen groups, mostly, at schools like Stanford, MIT, Harvard, Rockefeller University, most recently Johns Hopkins and Duke, growing and basically focusing their hiring of their academic professors into people that also think commercially, like you do. What is the climate in the Greek academic system today? Do we see people at your university or at the universities that you go around the country lecturing with professors like you that have been trained abroad or even just here and are thinking commercially? Are people starting to become more entrepreneurial in the Greek academic system? Not at all, better than 2012. What are your thoughts on that?
Oh, yeah. Okay, so let's start with the good news. It's definitely better than 2012. Definitely,
yes.
Definitely. I mean, there is
I added that because I saw your face
good.
And I wanted to give a positive message.
Yeah. There is no doubt that it's way better than it was back in 2012. We wish we had this kind of groups, at least from my point of view, in Greece, that they spin out like 10 companies a year. There are some groups at Harvard or MIT that they have like 5 companies or 1 company a year. They have huge rates of spin-off companies. There is no such thing here, but on the other side, there are great ideas. So in also my lab, we have like some rings that it's like a lateral flow, but instead of having like a strip, you have a ring. There is other labs that they have like apps that you cough, or Professor Nikita, that you can monitor your voice and you can predict COVID or not. Other labs that they're doing a lot of 3D printing for face masks and all these kind of medical devices. So
how many companies have grown though, Leonida? The question is, if I ask you right now to name 5, not 5 research projects, if I ask you to name 5 companies that colleagues of your fellow professors around the country have started, can you name 5?
Not in general in Greece, yes, I can name 5, but not 5 per university. 5 per the whole system, I can name 5 good ones. I mean, there are way more, but yes, I can name 5. But I hope that with this kind of mentality that is changing dramatically, if we have this discussion next year, we will find the 5 companies.
Absolutely, and that's the goal. That's why we are asking those questions, and you're giving those answers to get more people excited.
Change a little bit. I mean, this is a totally different topic, but we should have also a framework that is not so much bureaucratic, and you can spin out companies easier. And I know that previous government, but also this government as well. They're trying to have that kind of frameworks, so you can spin out companies easily. You can have technology licensing offices in all the universities that was not existing before.
Wow.
So all these things are changing.
That was my last question. If you thought that the existing administration, you also mentioned the previous one, have been helpful in making this process faster. You know, I have a lot of.
No, I'm talking both administrations. I'm not. I'm not. Of course, this administration, and we've been discussing with this administration of how to change in the legal framework. They are doing an incredible job, and the mentality have to change a little bit more, but it's definitely on the right way. Definitely. No.
Absolutely. Leonid, I'm really enjoying this discussion, but I have to switch to questions because we're a bit off time. And if that's okay, so I'm going to start taking questions from the crowd right now, and we already have a couple posted. One which I was planning to ask you, but I was worried some of the time, and now it's coming back. Let's talk a little bit about your COVID test and how far is the COVID test from commercialization, basically.
We have done the clinical performance. We have very, very good data, 99% and 95.8% sensitivity and specificity. And all the paperwork is ready. We just need to upload it to EOF and to get the CE mark. So within a few days, or not weeks, we're going to have the final product on the market.
And the EOF is like FDA in Greece, for those that are.
EOF is like the CE marking is the EMA, is the Greek.
The equivalent of.
But the equivalent, not the European equivalent, right? Because if it goes through EOF, it goes in whole Europe. So that's a big advantage.
The European equivalent of FDA for those of you that are in the US. Okay, good. We have another question. So do you see a growing market for diagnostics that focus on post-COVID comorbidities, where you focus away from virus antigens?
That's a good question. I see it's from Maris Persidis.
Are you friends?
Amazing. I know him, yes. And every time that I
good.
have a question, I just go to him. He was the ex-president of Hellenic Biocluster. So there is definitely a market for diagnostics. The only thing is that a lot of companies, way more than I would never thought, are doing the same thing. So there is a big market, but now the competition will show who's gonna win.
Very good. There's a lot of questions coming. 1 second. I'm trying to find the one that is more related to exactly what you said. So let's stay on the COVID topic. I'm going to take the COVID questions first. Everybody typing right now. So a question: what probability do you place on the development of an effective, persistent vaccine for COVID-19? Just to stay on the COVID topic for a bit.
Having worked with big vaccine companies, I feel it's highly probable. But I have to say that I'm not. I'm an engineer. I'm on the technology front, how to develop assays, so I'm not a virologist.
Yeah, Leonid, for everybody, Leonidas focuses on the detection side, not so much on the vaccine. So any vaccine questions, I mean,
fine, but I'm sure that they're going to develop a good vaccine. But it's just my personal opinion, and not a scientific one.
Yeah, for sure. And you know, follow-up question again on this from Vasily. Talking about the multiplexing data points, can you increase the throughput in any way so you can test more people faster?
Yes, so the multiplexing means that you can measure a lot of things in parallel. You can measure many antigens in parallel. You can measure many people in parallel. So usually those tests are taking place in 96-well plates, something like that. But in our case, usually you can measure only one thing in one well. In our case, you can measure up to 100 things in one well. So it's like having a stack of 100 plates, and you can measure that in 1 hour. So depending if you are multiplexing in parallel people or antigens for COVID, you can increase the throughput tremendously.
one final question on COVID, and then I'm going to switch back to the entrepreneurial ecosystem in Greece and to Newcos. What are the barriers to scale such antibody tests to entire countries that the non-science community fails to understand and appreciate?
We can scale it up. I mean, there are many different methods out there. Our biggest advantage is the multiplexability, so we can measure a lot of things simultaneously. And the scaling, the point that we have to make clear, it's not only the technology that can help scaling up. There are a lot of, it's the whole ecosystem that you have to, who's going to collect the blood? If you're going to collect it by people that go and travel with vans, with cars and collecting blood, whether you're going to do it on a punch card. Our method is very probable, and this is something that they do it in Sweden and have done it successfully. They use blood drops. So in a piece of paper, you put a blood of drop with a fingerprint, and you can mail it. And then there is a central lab that tests everything. So there is a lot of bottlenecks, and sometimes the bottleneck is not how many tests you can measure per hour, but how many tests you can collect per hour all over Greece.
For
so
sure.
It can be we have a different method with buccal. So from the cheek, you put like a swab, and you can scrape your cheek, and you can measure it with lower accuracy, but still good enough, depending on who's going to be the end user. You can do it from a blood spot. Dry blood spot is the name of the assay. So there are many different ways, but you need a consortium of many different expertises. Our method, and maybe I would like to finish with that, if there is someone on the consortium that is doing microfluidics, it's very amenable to microfluidics because it's a bit-based assay. So it can be a very easy, what we call point-of-care system. And because there is also a call in a few weeks, in 12 days in Greece, if someone from the audience knows like a point-of-care, a good point-of-care system that is amenable to bit-based assays, maybe we can talk later on.
So that's great. I'm not sure if I'm supposed to do this, but there's a question for me here, so I can ask it and answer it, I guess. The question is, do foreign VCs, I guess like us, consider following up the investments of Greek VCs if promising? Very quickly, I think that, great deals don't have, I had that conversation recently, great deals and great entrepreneurs is not about the country. It's about being a great deal, a great technology, and every good VC should be always open to do so. Specifically to Greece, I think that there's a bunch of exciting groups that have been started in the last 5, 6, 7 years, a little bit after Protavio got started. But on the immediate field of life sciences and biomedical innovations, which is where I focus, I don't think that yet Greece has a sophisticated group that understands drugs, devices, diagnostics, and global scaling today. And Leonidas, some people would love to talk to them. You're there, I'm not. But I always make a point when I am in Athens to spend time with companies in the life sciences world. We have a couple really good scientists. We have a couple really good entrepreneurs in the biotech space. I'm always interested to follow up with those, and I'm always interested in suggestions. To follow existing VCs in the biotech space, I don't think there are that many, or I don't think that we yet have grown to that level of having dedicated funds that understand drugs, devices, diagnostics as well as funds do abroad. So I think we have some way to go there on what Leonidas talked earlier about having a real infrastructure, a real ecosystem on the immediate life sciences community. We're working towards that. We're not there yet as a country on the VC side. But again, we don't need to just follow other Greek VCs. We can just meet directly with entrepreneurs and people like Leonidas that have excelled in what they do. And we're always excited to look at great deals. It doesn't matter where you're based, if you're in Greece or Australia or the US. So thank you for that. A question a question from Michael here. Which reforms do you need from the Greek government? I think the question is in regards to new cos and starting new companies, or I think it's related to that.
I think I can take this question. This is a great question. Personally, when we started this company, we started in 2012. We tried with a patent between, actually that was a patent between National Technical University of Athens and MIT. So we started in 2009 with the patent. The problems that we had, and there is a lot of room for improvement, is that there is no good technology licensing offices. There are not a lot of experts that they know how to take something from research and translate it. So you need a bottom-up approach. You need a very easy system. So someone that has an idea, he can easily create it. He can easily find access to a technology licensing office. He can easily patent something, either himself with the university or to give it to the technology licensing office. And an easy set of how you're going to share royalties, how you're going to share who's going to own the company, and to be something that is easily done and does not require a lot of meetings and a lot of decision from different policymakers. So from outside, we tried that for 3 years, and at the end, the company was not a startup, a spin-off company from the university. It was a simple startup company. Because back in 2011, we had spent 2012, for 2 years, we had spent a lot of time and we couldn't succeed on that. So I think the turning point for enabling translational research, it's a legal, an easy framework, mostly focusing on the technology licensing offices that can, that can write a patent within 1-2 weeks. They can submit, take decisions fast, and that's about it.
Thank you. Let me see how we're doing on time here. So one more question. We will take this question, and then I will ask you for some closing remarks to be respectful of everybody's time. So this question is very interesting, actually. What is the role of robotics and automation in streamlining testing and detection systems? I like this question.
Yeah. It has a central role, actually. These systems, I've seen a lot of systems. Most of the engineers, they don't know. It's very easy to make things that they are common in your life. Like if you're an engineer and you would like to develop things that you know and you know well, it's very easy to think of new ideas. But because you don't know all this pipeline, how the samples are collected and how you can, what's the legal framework to give the answer back to the patient, I think there is plenty of room for automating this whole thing of robotics. And I'm not talking only taking a vial of drop or a vial of blood and give the result, but also how you're going to collect it, how you're going to trace the blood all the way to give the test, to give the results back to the patient. So from the collection to the very end, to the giving the test back to the patient, this automation is very critical, and there is a lot of room for improvement.
Thank you. That's great. So with that, I would like to ask you, Leonida, do you have any closing remarks you would like to make? I know we talked about a lot of things.
I think it was a great discussion. Thank you very much for the introduction. If there are any other follow-up questions, you can Google me and you can connect with me. And thank you so much for having me here.
Leonida, thank you very much for joining us. We really appreciate it. I really enjoyed the discussion. And on a personal note, I've really enjoyed getting to know you and learning more about the Protavio story. I want to personally, as you know, follow up with you offline, following up our conversations the past weeks. Congratulations on what you and Sophia have accomplished. Very impressive. And again, thank you for your time. We hope everybody on the call appreciated hearing Leonidas give his perspective. And also, I want to direct your attention back to the screen. We'll have a similar talk again next week. It's not going to be on Monday because of Memorial Day weekend, I guess, in the US. It's going to be on Tuesday this time, May the 26th. Same time, same information, different speaker. We're gonna have Michael Bletas from the MIT Media Lab actually interview Alex Stamos, who was the chief security officer of Facebook, and before that Yahoo, and now he's the director of the Stanford the Stanford Internet Conservatory. My eyes are bad. So this is going to be very exciting. Alex is great. So I think you will all enjoy that. So with that, thank you for joining us, and we will see you soon next week. And please say hi to Leonidas' daughter as well.
Yeah.
Yeah.